Okay, so we've been talking all about evolution. We've talked about why it's important to know the evidence supporting the theory of evolution. Today we're going to delve a little bit deeper into one of those pieces of evidence, specifically fossils. Fossils help us to study the past. But what exactly is a fossil? The definition of a fossil is evidence or remains of once living plants or animals. Why do we study fossils found in rocks? To provide evidence of past existence of life forms. So as we've talked about before, this is providing evidence for evolution. It's providing evidence that organisms once lived here on the planet that are no longer alive, such as dinosaurs. To provide information about past environmental conditions. What type of environment were dinosaurs actually living in? What environment was required for their existence? And how is that different or similar to the present condition of Earth? To provide evidence that populations have undergone change over time due to environmental changes. This is what we know of as evolution. So here we have a picture of the Grand Canyon, and if you look at each layer of rock in the Grand Canyon, it's representing a different time period from Earth. And by examining those different layers and the fossils that are found in those layers, we can get a better understanding of what early Earth used to be like, or I should say earlier Earth. Again, looking at more rock layers. So let's take a deeper look at this idea. So there are different types of fossils that paleontologists, archaeologists are looking at. Original preservation fossils are fossils where the plant or animal remains have not undergone change since death. These types of fossils are very uncommon because what it means is that the organism became frozen, or the environment was extremely dry, or it was oxygen deprived. These types of environments prevent organic matter from beginning to decay, thus creating original preservation fossils. Some examples are mummified humans, frozen organisms such as Iceman, and mammoths and cats in the La Brea tar pits. Also fossilized insects in tree sap, as was popularized in the movie Jurassic Park. So here's an example of mummified human. The most commonly known culture for mummifying, of course, were the Egyptians. These were the artist's interpretation of what these tigers may have looked like before they became covered in essentially a type of tar that preserved them in their exact form. And then an insect in tree sap. The wonderful thing about original preservations is it really is preserving the organism in its original state, in an unaltered state, so that scientists really have a chance to know in great detail about the organism. Another type of fossil are altered hard parts, where all of the organic material has been replaced by rock. So basically what happens is minerals seep slowly in and replace the original organic tissue with silica, silolite, or pyrite, forming a rock-like fossil. So the original shape of the organism is preserved, but the original tissue is been replaced with other minerals. So the fossil has the same shape as the original object, but it's chemically more like a rock. So some examples of this are petrified wood, recrystallized shells. What we're seeing in this image here, this is petrified wood, and then these are recrystallized shells. So both of these were living organisms at one time, and they pretty much maintain the original shape, but chemically they're more like rocks than the original organism. All right, so molds and casts, which are common with shellfish. A mold is an impression or a cavity of the shell of an organism, and a cast is basically a cavity that might later become filled with minerals or sediment. So kind of think of it's kind of like baking a cake, where when you're baking this cake, you can pour it into a cake pan, and sometimes the cake pans actually come in images, like I've seen some that are in the shape of dogs, or another common one is a bundt cake pan, and you pour the batter in and you bake it, and when you're done baking it, you remove the cake from the pan, and the cake retains the image of the pan. Well, that's kind of the same idea of mold in cast fossils. What we see down here is how a cast actually forms. So the original organism, it dies. It gets buried in sand, sediment, whatever is present in the environment when it dies. And eventually, over time, that shell begins to be dissolved by groundwater, by the minerals that are present in groundwater. And the impression that is left behind eventually becomes filled up and creates sort of a mold, a cast of the organism that was once alive. Trace fossils are evidence of life other than the organism itself. So it's basically providing information about how an organism lived, moved, or obtained food. So some examples would be worm trails, burrows, and also footprints. What you're seeing in this image here, this is essentially a worm trail that is going into a burrow that's been preserved as a fossil. These are footprints. This is just a mechanical pencil that is laid here to give size to these footprints. Index fossils are used to define and identify geological periods. So basically, layers of sediment may look different, but contain index fossils from the same species and thus are from the same time period. So if you remember the layers of the Grand Canyon, that image that we saw at the beginning of this lecture, those layers, sometimes one layer can look quite different because it's composed of different minerals. But if fossils are examined from that same layer and the species that are fossilized in those layers are determined to have existed during the same time period, then scientists know that that layer is from the same time period, even though it's composed of different elements. So the specimen that has been fossilized in the layer of sediment, it has to be easily recognized. It has to be very abundant and widely distributed geographically in order to provide an index of time. It must have lived during a relatively short period of time, because the longer that a species lives, the harder it becomes to pinpoint the particular time period during which that layer of sediment would have formed. So some examples, most fossil-bearing rocks formed in the ocean, thus most major index fossils are marine organisms. This mollusk has a very distinct shape, and it's often used as an index fossil because its distinct shape helps it to be easily recognized. It was very abundant, widely distributed geographically, and it only lived for a short time period. So this mollusk makes an excellent index fossil. All right, so those are the different types of fossils that are used to pinpoint certain time periods, to know what organisms were living during those time periods and what type of environment the organisms need. But how do we actually date fossils? Dating rocks and fossils by placing them in chronological order without exact dates. So it's basically like saying that I'm older than you. Well, we know this to be true, but that doesn't actually tell you how old I am or how old you are. It just gives us a relative age dating. So the law of superposition is a law that in an undisturbed sequence, the oldest rocks are at the bottom and each successive layer is younger. So again, if we look at those layers of the Grand Canyon, the layers that are going to be at the top are going to be younger. The layers that are at the bottom are going to be older. The principle of cross-cutting relationships, if there is an intrusion or a fault in a layer of sediment, that intrusion or fault is younger than the rock that it cuts across. And these intrusions are quite common in areas where there are earthquakes. And then correlation, which is matching rocks and fossils of one geological region to another. So correlations are used to date rock layers that are far apart from each other geographically. And geologists examine rocks for distinctive fossils, again, those index fossils, and other features to help them identify and date these different layers. So that's relative age dating. It doesn't give us an exact time period, but it gives us an idea. So relative age dating, if we look at this image here, if we look at these different layers, we can see that A is on the bottom and I is on the top. Well, according to the law of superposition, A is the oldest and I is the youngest. But we have a fault here, so this probably formed in an area where there are earthquakes. So rock layers on the west side of the outcrop do not match layers that are on the east side because of this fault and this intrusion. So which is going to be younger, layer A or layer C? Well, according to the theory of cross-cutting relationships, it's still going to be C, because the layer that is cutting across is the layer that is younger. Absolute age dating. Absolute age dating is used to determine the actual age of a rock, fossil, or other object. So we're not just saying that, well, this fossil is older than this fossil because it was found lower down in the layers of sediment. We're actually finding an exact age, and some methods that are used, tree rings. So each ring represents 1 year of growth. So if you count the rings on a tree, you'll know how old the tree is, exactly how old the tree is. Varves are bands of sediment that show a yearly cycle from climate change. So although both of these are accurate means of finding an exact age, neither can be used to date very far back in time. Trees don't live that long. There has to be another method of determining age. And in fact, there is. This method is radioactive dating. Radioactive dating is dating fossils based on the amount of radioactive material that is remaining in a substance over time. So to understand radioactive dating, you need to understand a little bit about atomic structure. So basic atomic structure, atoms consist of a nucleus, and that nucleus is made of protons and neutrons, and orbiting the nucleus are electrons. The proton has a positive charge, the neutron has no charge, and the electron has an electric charge. So that's the basic structure of an atom. Now, isotopes are an atom with an abnormal amount of neutrons. This makes atoms unstable and they may begin to break down. So in a stable atom, there's an equal number of protons to neutrons. In an isotope or an unstable atom, there is an uneven number of protons to neutrons. So when we look at carbon-12 is a stable atom because it has 6 protons and 6 neutrons. Carbon-13, it is in fact an isotope because there's 1 more neutron than proton. However, it's still considered pretty relatively stable because the difference isn't great enough. There's a lot of force holding protons and neutrons together, so there has to be a significant amount of instability to cause an atom to begin to decay, which is what we see with carbon-14. With carbon-14, there are 6 protons and 8 neutrons. This difference is enough to make carbon-14 radioactive. And this is very, very important because carbon-14 is what is used to determine absolute ages of fossils that were once living. Let's take a closer look at this idea. All right, so the process of radioactive dating, how it actually works. Radioactive substances, also known as unstable atoms, are emitting protons and neutrons at a constant rate. So because that nucleus is unstable, it's actually trying to get rid of protons and neutrons so that it becomes more stable. Atoms are always trying to be stable. So when they're unstable, when they're an isotope, they begin to break down so they can become more stable. Another way that atoms become stable is by bonding with other atoms. That's why chemical reactions happen. So as the number of nuclear particles changes during decay, the original element, which is known as the parent atom, is converted into a different element, which is known as the daughter atom. So essentially, carbon-14 is going to be breaking down into another element. The rate of emission remains constant regardless of environmental changes. So the rate at which that atom is emitting protons and neutrons remains constant regardless of environmental changes. Since the rate of decay is constant, you can measure the parent-to-daughter ratio to determine the age of the rock or the organism. Half-life is the length of time it takes for 1/2 of the original amount of an element to decay. So looking at this graph of carbon-14, if we started out with 10 grams of carbon-14, one half-life is going to be when that 10 grams is reduced to 5. 2 half-lives is going to be when that 5 is reduced to 2.5. 4 half-lives is going to, 3 half-lives is when the 2.5 is reduced in half to 1.25, and so on. So it's always half. This is why it's called a half-life, because it's always half of half of half of half. So what that means is that half-lives occur at an exponential rate. It starts out quickly and begins to slow down more and more over time. So carbon and radioactive dating. Why is this so important? Why are we learning about carbon-14? Because all living organisms contain both carbon-12 and carbon-14. If an organism is on this planet and it's alive, it contains carbon-12 and it contains carbon-14, which is great for archaeologists and paleontologists because they can measure the rate of decay of carbon-14 in order to determine the age of a fossil. So even though carbon-14 slowly decays, it is continuously replaced when an organism is alive so that the ratio of carbon-14 to carbon-12 is constant when an organism is alive. So basically what this means is you take in carbon-14 when you eat food, you get rid of carbon-14 when you poop, which creates a constant ratio while you are alive. Now, when you start to die, you're no longer taking in food, you're no longer taking in carbon-14, so that carbon-14 slowly begins to break down into carbon-12. When an organism dies, carbon-14 is no longer replaced because the organism is obviously no longer taking in energy, and the constant ratio of carbon-14 to carbon-12 decreases. An estimate of the amount of time that has lapsed since death of the organism can be found using radioactive dating to determine the amount of carbon-14 found in dead tissue. Another example is uranium. Uranium breaks down into lead. So, and you're going to want to draw this image on your notes. So, if no time has passed, then there's 100% of uranium present. After one half-life, there's only 50% uranium and 50% lead. The half-life of uranium is 4.5 billion years, quite a bit more than carbon-14, which I believe is 5,000 or 7,000 years. After 2 half-lives, how much uranium is going to be left? Well, 25%. 25% uranium, 75% lead. And that's going to be after 9 billion years because one half-life for uranium is 4.5 billion years. So a half-life is half of the original amount. This is what it looks like on a graph. So the percentage of uranium remaining is on our y-axis and time is on our x-axis. So time is our independent variable and the percentage of remaining uranium is our dependent variable. And this is what we can see. It's an exponential decay. It starts out very quickly, but then begins to decay much more slowly because there's less and less uranium that needs to be decayed. All right, if you want to take a closer look at this, you can click on this link and it will actually take you to a simulation where you can see that exponential decay. You can see an element decay very, very quickly, and then the decay rates start to slow down depending on the half-life. All right, so that's dating fossils. That's the different types of fossils that are used to date, how we date those different fossils using both absolute age dating and relative age dating, and the importance of carbon-14 and absolute age dating, because carbon-14 is found in all living organisms.